We found that they have a molecular signature which is very similar to the molecular signature of fully developed tumors, but what's unique about them is we can identify them in patients sometimes several years before the patient eventually develops a tumor. My name is Dr. Rodrigo Carlessi, and I'm a senior research fellow here at the Curtin Medical Research Institute at Curtin University in Perth, Western Australia. My career started back in my hometown in southern Brazil. I did an undergraduate degree, a bachelor's in life science with a major in molecular and cellular biology. When I finished that, I went to Israel and I joined the Weizmann Institute of Science, and I've obtained a master's in molecular genetics. I then went back to Brazil and joined the medical school of the Federal University in my hometown, where I did a PhD in molecular endocrinology. As part of my PhD, I was also able to be awarded a scholarship from the Brazilian government to spend 1 year of my research abroad, and I chose to come to Australia. This is when I first came to Perth. I did this 1 year as a visiting student here, and this is when I decided I wanted to come back to Australia for my postdoc. And so I just went back to Brazil, finished my PhD, and joined the then School of Biomedical Science here at Curtin University. And I did my postdoc working primarily with beta cell metabolism. Beta cells are the pancreatic cells that produce insulin, and my research was focused then in diabetes. I then, from this position, I was then recruited to take up the position that I'm currently on as the lead of the Cancer Genomics Group within the Liver Disease and Regeneration Laboratory here at the Curtin Medical Research Institute. In my group, we primarily focus on utilizing cutting-edge genomics technologies to address research questions in liver cancer. My primary area of research is liver cancer. Liver cancer, among all human cancers, has one of the worst survival rates, with only about 20% of patients surviving 5 years after their diagnosis. And this really low survival rate is attributed to a number of factors, but primarily two things. One of them is the therapies for liver cancer are not highly successful. Even with recent progress that has been done with the advent of immunotherapies for liver cancer, it's still only a small fraction of patients, about 25%, respond well to these therapies. So one area of research is actually developing better therapeutics for liver cancer. And the other area that also drives this low survival rate is the fact that liver cancer is mostly diagnosed at stages of the disease where there is no curative approaches. Despite the fact that early liver cancer can be cured, most patients, about 3/4 of them, are unfortunately diagnosed at the advanced stages. And therefore, if you can diagnose liver cancer early, this would be one of the best ways to promote better survival. So this is another focus of our research here. We develop diagnostic assays to be able to detect the disease in the early stages. We are very heavy on a set of diagnostic assays that we call liquid biopsies, where we use genetic material. We use the ability to identify genetic material from the cancer in the blood as a way to be able to, diagnose the disease at early stages. And we also try to use these liquid biopsy assays in order to better monitor the disease in those patients who have already been diagnosed with the disease. And this way we can better monitor the progression, how these patients respond to therapies, and so on. One important turning point in my career was when my group and I were able to identify these premalignant liver cells. We employed a technique called single nucleus RNA sequencing. And we then first identified and then deeply characterized the cell state in the liver of cells that accumulate mutations over the course of chronic liver disease. And we identified them as the source of liver cancer. Now, we found that they have a molecular signature which is very similar to the molecular signature of fully developed tumors, but what's unique about them is we can identify them in patients sometimes several years before the patient eventually develops a tumor. Now, in parallel with the discovery of these cells, we started a collaboration with a group from Monash University, led by Professor Mark Fabraio. And collaborating with Mark, we're able to test a number of therapeutic approaches in order to prevent the accumulation of those cells. And we end up finding that the combination of compounds that treat or that inhibit inflammation as well as block endoplasmic reticular stress, we're able to see that some of these compounds were able to reduce the accumulation of these premalignant cells. And in parallel with this observation, we saw that the mice treated with the compounds also had a remarkable reduction in the incidence of liver cancer, showing that if you can target these cells early on, this is one way to prevent the development of tumors. Lately, we also started a collaboration with a group from University of California, San Diego, led by Professor Michael Caring. And this generated or resulted in a high-profile publication in Nature earlier this year. This is a study where we were able to, for the first time, demonstrate a molecular mechanism that basically licenses these premalignant cells into converting into fully malignant. We identified FBP1, an enzyme which is normally associated with metabolic pathways. It's involved in the gluconeogenesis pathway, but we found that it also works as a tumor suppressor in the liver, and these premalignant cells, they eventually lose the expression of this enzyme, and this is mostly due to a hypermethylation of the promoter of the gene. And we saw that when the tumor suppressor is lost, this is what licensed the conversion of premalignant cells into fully malignant cells. We have had success in securing funding from diverse funding organizations over the years, including NHMRC, which is a national body that funds medical research in Australia. We have also received funding from not-for-profit organizations, including the Cancer Council of Western Australia. And more recently, it's probably most of the current research we're doing is funded by the Cancer Research Trust, an organization that funds cancer research here in Western Australia. And thanks to funding that we've received from the CRT, we're able to establish, along with many other colleagues in the field, a large collaborative for liver cancer research here in Western Australia called the Liver Cancer Collaborative. My passion has always been in trying to understand complex biological processes and mechanisms in life. In the last few years of my career, I have almost exclusively dedicated to research in the area of cancer, particularly liver cancer. And then researching in this area also brought me another passion, which is basically to be able to work with something that can make a difference for the life of people. I have a family with 2 little girls, one 5-year-old and one baby who is turning 1 year now in the next month. I love to spend time with them when I'm not working here. I have always been a fan of water sports, and one of my favorite ones has been kite surfing. So I absolutely love to go out there here in WA in the summertime where we have a really good sea breeze, and I go out there with my friends and have fun.